Okay. All right. Well, I want to welcome everybody to the awarding of the SIAM Life Sciences Activity Group Early Career Prize lecture. And I'm Victoria Booth, the chair of the Life Sciences SIAG, but I'd like to introduce Linda Allen, who was chair of the nominating committee for the early prize, to do the presentation of the award and introduction of our award winner. So go ahead, Linda.
Thank you very much. So as a representative of the selection committee and the Life Sciences Activity Group, it is my pleasure to give the Life Sciences Early Career Prize for 2020 to Kevin Hannay. Kevin received his PhD in 2017 from the University of Michigan under the supervision of Daniel Forger and Victoria Booth. Currently, he is an assistant professor at Schreiner University. Kevin's research makes a significant contribution to both mathematics and life sciences, which is evident in his key 2018 paper, Macroscopic Models for Networks of Coupled Biological Oscillators in Science Advances. The award reads, the SIAM Activity Group on Life Sciences presents the Early Career Prize to Kevin Hannay for his major advancement of coupled oscillator systems through identification of a new macroscopic reduction that captures properties of phase distributions observed in experimental data. Today, Kevin's presentation is Towards Personalized Models of Human Circadian Rhythms. Kevin?
Well, thank you very much for the award and for all the time that you've put into this. And setting this up, and you know, I really appreciate the recognition, and I'm very humbled by the honor of receiving this award.
Congratulations.
I mean, it really means a lot to me. So thank you. Yeah, and so I'd like to tell you just a little bit today about some work that I've been doing on developing personal models for circadian rhythms. So circadian rhythms are the body's natural 24-hour cycles in physiology, the most obvious of which is sleep-wake, but almost every physiological process in the body shows a 24-hour rhythm. So like blood pressure or body temperature, melatonin secretion, and you know, just about every, I could keep listing things, but you get the point. So this is essential to you know, a healthy, maintaining homeostasis throughout the body. And when they become disrupted, then tons of different things can go wrong, both on a you know, physical health, but also mental health standpoint. And in mammals, the main way, you know, by going wrong, we mean that your internal rhythms become desynchronized from the environment. And so how do you stay in, you know, how are we supposed to stay in sync with the environment? Well, in mammals, the main cue is the external light cycle. In the natural system that the circadian rhythms evolved for, we've got our light coming into the circadian system, and then that's influencing our behavioral cycles. And so, you know, it would be crazy to think that my behavioral cycle was somehow affecting the solar cycle, right? The sun doesn't care when I get up in the morning, or, you know, doesn't care about my behavior. However, so that's what the system that the circadian rhythm evolved for. Modern life is very different. So the invention of artificial lighting has allowed our behavior to feed back onto the system. And so now we can choose to drive the light input, which you can imagine causes all kinds of potential for disruptions and things. Not to mention the jet lag, you know, when we actually do change the solar cycle by traveling quickly across time zones. But just on a day-to-day cycle, our choices can now influence this. Okay, so from a biological standpoint, a lot is known about this circadian rhythm and what controls it. So in your brain, there's a small region called the suprachiasmatic nucleus, or SCN. And inside of that, there are like 20,000 clock neurons. And so those neurons each have a very intricate biochemical cycle that runs with a period of about 24 hours. This is the awarding of the Nobel Prize for working out the molecular details of this cycle. And so each one of those cells is then coupled together, and they produce a collective rhythm that drives your behavior. So it's kind of a rare instance in the brain where we have the one region driving a behavior, organism level behavior. Usually it's a bunch of regions interacting. So this is a very interesting and challenging problem from a mathematical standpoint because of the multiscale nature. So we have time scales of anywhere from the milliseconds for the neurons firing all the way up to months, weeks, months, years for changes in the behavior. And it's a very high dimensional problem. So if we were to incorporate all that is known, we'd have something like 10 to the 11th parameters to fit if we want to model human dynamics. So obviously we don't have enough data to fit 10 to the 11th parameters. So the approach that we took is to take a high dimensional model and kind of coarse grain it and hopefully in a systematic way down to a low dimensional macroscopic model, and then fit the parameters and use that to make predictions. So here's a high dimensional model for human circadian rhythms. So we've got our phase oscillator. So we just described each clock neuron with one phase variable. It's heterogeneity, noise. The sum here is a coupling, and this last term in 1A is the light input. And Q is how each cell reacts to the light input coming from the eyes. So this is still relatively high dimensional. It's about one for each neuron, so about 10 to 20,000 equations. So the real motivation from this came from looking in the coupled oscillator literature at this technique known as the Antonsen reduction, or Antonsen technique. Ansatz, it's got many names. It's very important, so it's got so many variants or remixes. But the idea is that you can take like a Kuramoto oscillator system of heterogeneous oscillators and reduce it exactly to a macroscopic system. And so you can see like the K here is the same here. So all the parameters and variables can be traced back to that high dimensional system. And so I was, as a graduate student, I was very interested in whether I could apply this to study human circadian rhythms. So there's a few potential issues here, right? So a technique that's that powerful is going to come with some assumptions. And so one of them is a practical one in the heterogeneity should follow a Cauchy distribution, or Lorentzian distribution, as the physicists call it. The coupling should be relatively simple and all-to-all, and no noise is allowed in the formal Antonsen reduction. So we translated that. All of those assumptions can be written in the form of like a moment closure on something called the Dietoorder parameters, which just express a property of the phase distribution. And so we then looked for that signature by extracting some phase distribution data from mice SEN. So we took some data that was collected in the Herzog lab, and where they extract a whole SEN, culture it, and monitor the phase of thousands of neurons over a long period of time. And so in this experiment, they culture it, and then they add TTX, which blocks all the transmission. So all the neurons desynchronize, and then they monitor the resynchronization. And so this gives us a good measurement of what the phase distribution of neurons in the SEN might look like. So if you take those and you extract it, and we plot, so each green point up in the first row here is a time point in that time series. And we're looking for, so if it obeyed the Antonsen closure, it would be along the dotted line here. And we can see that actually it converges pretty quickly to a different relationship between those moments. And so after some analysis and looking at it, we found that it's actually R1 to the m squared. And so this means that those moments fall off more quickly than would be predicted by the Antonsen technique. So, you know, it's sort of yada over the details of the reduction, but really you can substitute in that moment closure for the Antonsen one and follow a pretty similar procedure. We can take our high-dimensional model and systematically reduce it now to a two-dimensional macroscopic model, for R being the amplitude, collective amplitude of the SCN, and psi being the mean phase. And all of those parameters actually mean something as far as our microscopic model. So now we're at the scale where we can actually use some of the data for humans to fit this. So we took phase response curve data, which measures, given an impulse, given a light pulse for humans, how does that affect your circadian rhythm? So like if I give you a light pulse at 5 a.m. or something, then that if you're well and trained and you're sleeping, then wake you up and shine it in your face, that's going to give you a phase advance. So it'll move your rhythms forward by some amount for that day. Conversely, if I do that like right before bed and you get like bright light, then that will delay you and it'll be hard to get to sleep for a while, basically. So if you trace that out over a whole cycle, you can trace out what's called a phase response curve. And so that's the data that we use to fit that low dimensional model. So we've got 14 parameters. We put a least squares cost function on it and fit. So this is the training data for the model. The green points are the experimental data. The blue is the best fit curve for a single parameter set. And the green cloud is a Markov chain over, so in this case, we're looking for all parameters that are consistent with the data, or so a posterior density for the predictions. So that gives us a posterior for each parameter. And so the question that we're now looking at is, well, how much does that matter, right? So we spend a lot of time getting parameters that we can interpret and things by deriving this. So do those parameters actually matter in the wild, right? So we can take those parameters and just run them. So here I took a sample of 30 parameter sets that are consistent with the PRC data and ran it through a Jetlag protocol. So you read this as a light schedule, times increasing down. So we have a sudden shift in the light schedule. I think it's like an 8-hour shift eastbound. And so we're moving down and then all of a sudden there's a shift. And so these are two different phase markers. So just they're the same, you're at the same period in your clock at each point in these days. So you can see when you're in train, then it occurs at the same time every day. You're having a consistent rhythm. All of a sudden there's a shift and it has to adjust. And you can see that among the parameter sets that are consistent, we can see different behaviors here, right? So in this case, we're adjusting in one direction versus the other, which can then would affect our predictions of what the best way to mitigate jet lag or something is. This gets especially bad if I look for something like a shift worker that works a 5 days on, 2 days off kind of shift schedule. So once again, taking some parameters that are all consistent with the data that we have so far, we can see, this just is supposed to look like a big mess if it looks like a big mess, because the predictions are sort of spreading out and diverging pretty quickly here, right? So this tells us that the parameters, in fact, do matter, right? It's going to be very hard to generate accurate predictions from the model if we don't know what the individual's parameters are, at least under some of these light schedules. So the hope is that we can use some data that's big data, can be the cure for this. We're using these wearable devices that give us sort of long-term time series of these estimates that maybe are noisy themselves. Oh yeah, and so I'm trying to get this going to call this a chronoprint. Instead of a fingerprint, it's your chronoprint is your parameter set. But we'll see if that catches on. I'm just hoping. Okay, so just some examples of trying to recover these for simulated data. So if I take, we're just going to try to fit this using a maximization for the posterior. So we get a map estimate for our parameters. So for example, if I look at just regular light conditions, so the same schedule every day, and I pretend that I've measured phase markers for 30 days, which is really long time, more data than we normally have, can I recover some of the essential parameters for the model from those observations? And and so we added noise and things to the simulations to make it more realistic. But one of the essential parameters is the intrinsic period of your clock, and that's known to vary among individuals. And you can see that even from this, if we have long enough time series, we can do a somewhat accurate recovery just using this sort of naive map, maximizing the posterior probability. But if we look at, say, jet lag, so now we've observed some transient dynamics instead of just around the fixed schedule, I can do much better on the period recovery. And I can even recover some additional parameters as well. And for a shift worker, which we saw was one of the most divergent cases, you can do extremely well in recovering those parameters. Which of course, I mean, so those two things are one of the same, right? So if we see different behaviors, then we should be able to fit the parameters. And if there's not essential differences, then we won't be able to fit them. So the two plots are related there. But the next step that we're working on is to really try to do this for experimental data, in which case we'll have much less accurate measurements or high fidelity measurements of their circadian phase, but we can potentially have long time series of noisier data. And the other challenges here are to start incorporating more of the demographic known quantities for estimating the parameters and feeding those into the model itself. Okay, so just one last note here, if I've got any time left, would be that, none of this at all was planned, or, so I present this, we always present our work as if we start at point A and we end up at point B. But like, I spent most of my time wandering around. So this is a random walk, and then I fit a line to it, right? So the talk is a fitted line, but really this took years of just stumbling around. I had no idea if any of this was going to work. I still don't, and so the only reason that I made any progress at all on this was because I had good advice and I had people supporting me. And so on that note, I'd especially like to thank my advisors, Victoria Booth and Danny Forger, who, if you make one important choice in your academic career, it's choosing your advisors. And I just got lucky because it's not like I knew that going in. And then my collaborators have also been absolutely essential in this. So I want to thank all those people that have supported me over the years. Are there any questions?
Oh, thank you, Kevin. Here, I'll clap for everybody. And so, yeah, so now we'll open up for questions. I guess, Suzanne, I think I'm still on my phone and it might be hard for me to see if somebody raises their hand.
I can see the chat and no one has asked the question yet. So
okay. So I guess, so if you have a question, either
we can unmute people if you want us to unmute some people. Yeah, if they have a question, let us know. We want to know. Maybe I missed this. Did you tell where some of this work has been published? Or I didn't hear. Maybe I missed that slide.
Oh, yeah, that would have been a good thing to include. Yeah, so the mathematical background was the Science Advances in 2018 paper. And this last October, we published the human model in the Journal of Biological Rhythms. So those things are on my website as well.
Okay, thank you.
But I will point out, if anybody wants to use the model, all of this is in a Python package that you can download and simulate and create these graphs for yourself. So that there's an open source way to quickly get up to speed on it.
I do have there is a question that's in the chat, which says, this is from Lizette. Can you say more about your parameter fitting and whether you've been able to look into parameter identifiability?
Yeah, yeah. So we're looking at the identifiability now. The parameter fitting. So originally we ran it as a long sort of genetic algorithm to move around the cost space, but I've recently sort of rewritten that to do more of a gradient descent and then random start gradient descent stuff. And I'm hoping to publish that with the Python package as well. Because as more data is included and published and things, then my expectation is that people will want to refit and get new parameter chains. But as far as the identifiability question, so I mean that's the technical term for do the parameters matter, right? So of course,
Or no,
in this
uniqueness,
case,
maybe uniquely identify or approximately uniquely identify, right?
yeah. So it depends on the light schedules, right? So we're working on that now, but that's a really important question.
Okay. There's another question here from Maeve McCarthy, which says that when you say 30 days of data is a lot, how much is typical? What kind of data is measured?
Yeah, well, so one, maybe two measurements. So the issue here, and people are working on pushing this out of the laboratory, but to get a phase measurement for human circadian rhythms like that, people have to come in and be put in darkness for a period of hours, and they measure when your melatonin starts to increase. It's called the DILMO, or dim light melatonin onset. And so in most of the human data that we have, we'll have recordings of their light and things for maybe weeks at a time, a time scale of weeks, and there'll be a couple of times where they come in and officially measure their circadian rhythms and estimate it. And so the other thing that's the untapped potential there is to try to use some of the sleep data that you can extract from wearable devices as a noisy estimate, but there's lots of confounding factors there.
Actually, I missed a question earlier, so I want to point this out. This is from Derek Orr. How did you find that R sub m equals R to the m squared and not RM like odd ant? So experiment, and they said question mark, experimentally?
Yeah, so originally experimentally. So I kind of looking at the data here. So I made this plot as a graduate student, and I was really hoping that it would match the R1 to the M, and it just didn't. But if you look at the Science Advances paper, what's going on there is that this is a circular Gaussian, basically, right? So the M squared is what you would expect if you have exponential tails on a distribution, and the R1 to the M is polynomial decay. And so there's some theoretical explanations for why you would see one versus the other. But the justification for circadian rhythms comes from experiment.
Okay, thank you. I didn't see another question. Did I miss it? Anyway, thank you very much.
Thank you.
Okay.
Yes. Thanks, Kevin.
Yes, thanks.
Yeah, I enjoyed it too, Kevin. Thank you much. Thank you very much.